First of all, Assalamualaikum and good afternoon, everyone, today. So first of all, I'd like to welcome you to our webinar this afternoon entitled NGS Next Generation Sequencing, Unleash Limitless Discovery on Oral Microbiome. Okay, very scientific, and then we sincerely thank you for choosing, to spend your lunchtime with us today. I'm pretty sure you know myself already, but it is a great pleasure for me to introduce our speaker today, who is going to share his experience with us on the latest application using next generation sequencing technology in facilitating oral microbiome related research in our fields mainly. So a little bit on speakers. Mr. Lim Lixin. He is an assistant manager in Genomic, with under sales department in science vision syndrome. He has 13 years of molecular experience. He also provides consultation service to several government research facilities such as Mahkamah Kesihatan Awam Kebangsaan, National Public Health Laboratory, Mahkamah Keselamatan dan Fasiliti Makanan, Food Quality and Safety Laboratory, not to forget Institute of Medical Research under National Institute of Health, and of course several other research universities on infectious disease detection and surveillance, currently on COVID-19, TB, dengue, HIV, and salmonella, to name a few. His vast experience gained him the recognition as master of genomic applications. Very exciting to listen to you today. He also helped in assisting IMR to detect the SARS-CoV-2 variants such as D614G and B117. So just listing out his achievement is already overwhelming, even for myself. So without further ado, let us welcome the man himself, Mr. Lim. The screen is yours.
Yeah, thanks for the chairperson. Can you hear me well?
Yes, probably a little bit louder would help, I think.
Okay, I'll try to speak louder. But if you have a difficulty in hearing, please voice out or you can post the questions in the chat box so I can address it later on. Okay.
Okay.
Let me get the screenshot. Are you able to see the screen?
Okay, sharing now, coming up, yeah.
It's a bit black. Okay. Yep. Okay.
Yes, we can
Okay.
see your screen.
Yep.
Yep.
Okay. Okay. Can. Okay. Yep. So okay. So my name is Lixin. Thank you for the chairperson for a really honor introductions on that. So yeah, it's my honor to be able to share with you some experience that I have throughout these years serving the research community, as well as looking into research and diagnostic or screening in the future. So today's title is looking at NGS, which the technology that can help unleash the limitless discovery on the oral microbiome. Okay, throughout these sessions, I'll cover first some of the agenda I'll cover today. So first I'll let you know, okay, who are we? Okay, what do we do? Okay, and then a brief introductions about Illumina NGS technology, which is what most of the people are using. Okay, and then quickly go through what are the metagenomics approach available. Okay, and then lastly, we'll give you some examples of the microbiome discovery which enabled by NGS. So this is not limited to this one, but just to give you ideas how powerful can the NGS enable for the microbiome study. So the content will be more focused on the oral related microbiome. We know that microbiome exists in a lot of places, and then they play a role any places that you can name it. So basically, but today the audience more focusing on oral, so I'll give you some of the examples of the oral related microbiome. Okay, so let's start. Okay, so first of all, I'll do just a quick intro on who are we. I mean, some of you may just be the first time hearing like who is Science Vision. Okay, and we actually established since 2007, and has become the one of the most reputable and trusted scientific solution provider in Malaysia. So at Science Visions, basically our we take the pride in becoming the catalyst to increase the technology adoptions by becoming the active link between customers and technology. So meaning that we will train you up on how to utilize the technology so that you can actually benefit from the technology to gain more, as well as having a more breakthrough in your research as well. Okay. Okay, so our aim is really to unlock the power of genome to improve the human health. And as mentioned, microbiome is very important for human. So I mean, looking at the microbiome is also part of the way to improve the human health. Okay, and with technology is very enabling. So it enables us to do a lot of things with this NGS technology, which I will talk more examples later on. Okay, so basically, who do we serve? We actually, I mean, as a start, NGS is just mainly focused on research, right? But I mean, we do see because of the enabling technology, you are seeing more and more adaptations from different fields of it, okay, such as listed here, the reproductive health, oncology, which is the most rely on the NGS because a lot of disease diagnostic are really based on the genome itself. Looking at population sequencing, research, which I'll go through you what are the research possibility enabled by the NGS as well, and some complex diseases like rare disease, and now also people are looking into consumer, which can tell you the health conditions of you, what are the informations that you can benefit from your genome itself, okay, as well as infectious disease. So as mentioned by the chairperson, sequencing has been very helpful to actually combating this pandemic, especially the COVID, where from the start to the end until now, is basically leveraging the NGS to identify the novel pathogens, hence provide the ground for the discovery and then detections and as well as a plan to tackle the pathogen. So it's also used in the forensic, like finding the ID to confirm the suspect and evidence, as well as to improve on the agriculture, what breed is better than the others, which are more cost effective or efficient, and then looking at diagnostic of genetic health, and lastly also archiving the bio data, okay, in the big data format, so that the biopharma can be more efficient in the work. Okay. Yeah, so in Science Vision, we provide a range of solutions, okay, but mainly focused on NGS. However, there's other solutions we also cater as well, which cover a wide range here, like including the life science, lab essentials, microarray solution, and also bioinformatics, which I'll touch a bit more why the needs of the bioinformatics. And we basically, Science Vision have a sister company which really dedicated on bioinformatics, because a lot of the times the researchers working on NGS have a bottlenecks of a bioinformatics. So here we basically streamline the solution so that the researchers can more progressive in their work. Okay, and also the common PCR and qPCR solution, as well as food safety solutions, leveraging the NGS technology. Okay. Yeah. So as mentioned, Science Vision have a sister company named Bioec, which really dedicated for bioinformatics, okay, helping researchers to overcome some of the bioinformatic bottlenecks. So here, these are amongst the solutions that are provided by Bioec, okay, which includes the trainings, okay, and hardware setup for your NGS work, data analysis for the NGS work, such as looking at microbiome, what are the significant findings, all those with the help of analysis, you basically can simplify the information capture and more conclusive as well. So we also develop software, okay, and this software we really developed towards the customer needs. And here, like on the food safety, as well as for the SARS-CoV genome analysis, which requested by Ministry of Health as well. And then for some surveillance works, looking into the AMR surveillance. So this is also one of the things that the areas that of a focus with BioInfo as well. So and also it's a collaborations. We do have collaborations work in place with researchers. Okay, if that's something potential or needed with the researchers. Okay, so in that sense, these are some of the bioinformatics solutions. If you come across or any challenges on your bioinformatics workflow in the NGS, please feel free to reach us. We do have a wide range of solutions that maybe can cater for your needs. Okay? Yeah, and we, as mentioned, we supply quite a number of solutions, and here are our our partners. Okay, we are working closely with them to provide the solutions to address the customer needs and also challenges. So here, including software solutions, instruments, as well as some reagent as well. It's a wide range as listed just now. Okay, and how do you reach us? Is that you can connect to us with Twitter, LinkedIn, or Facebook. So here are some of our. You can find us at here. And we basically represent, we are present in a few countries as well, in Malaysia, Myanmar, Brunei, as well as Nepal. Okay. Yeah. So, okay, so let's back to the main topic of today, so which is the NGS. So, as mentioned, NGS back in, I think they have like 15 years of history since I think 2007 when it's first available. So back then it's actually very costly as well. Although comparing to the Sanger sequencing, it's really it's still really much improvement. Okay, and then because of the availability of the next generation sequencing, so it's basically revolutionized how people do sequencing. Okay, and yeah, and so as you can see here, I mean, basically, the NGS is really a stepping stone for the sequencing technology, which is revolutionized, made a lot of the things, which previously the sequencing is only for research purposes, but because of the affordability, it has already reached out to a lot of the field, different segments, different markets as well in that sense. So it is even terms as like a law based on the founder, I mean, based on the CEO of the Illumina previously, which is called a Fedley law. So this Illumina is a company that's beating the genetic revolution. So if you look at the chart here, why they claim so is that before the availability of the NGS, which is before 2007, okay, you're looking at like the sequencing of one human genome is looking at a very like pricey manner where you need to, if you want to sequence a human genome, it takes about 10 million US dollar, okay, to get a human genome. And that, I mean, with available or Sanger sequencer, okay, although it's cheaper as it goes, where there's a term called Moore's law. So Moore's law is actually a law for the decrease of the cost for technology, okay, where like IT, okay, computers, right, as you go, right, the price will be reduced by half, okay, as time goes, okay. But then in NGS wise, right, it's totally different until like people have maybe a law because it's not following the common technology Moore's law. So back in 2007, okay, the sequencer genome is really costing like 10 million US dollar. But with the introducing of NGS, right, it exponentially reduced the price of sequencing to even like less than 1 million within a years of available of the NGS, and that continued to drop until like even within a few years of time, it had already significantly reduced to $10,000 US per genome, and that also continued to further reduce, right? When there's a new technology improvement from Illumina, where they continue to improve their technology and making a human genome even drop nearer to $1,000 US per genome. And back in today, right, we are looking at really less than $1,000 US for one human genome. And this is really very enabling, not only for human, but also for other animals, organisms, right, which do not have funding or challenges, right, such as like plant and animals, right, then we are able to know more on this organism. Okay, with that enabling, you can see a lot of applications is made available leveraging the NGS technology, and then there's more solution now are enabled for you to do the work or investigative work, and also more fundamental research is enabling. So making us like the human genome research or the profile of to very intensively study the human genome is very enabling, and there's a progressive over years and years. Okay, yeah. So this is actually with that enabling, right, is actually getting a lot of attentions to from different areas, okay, such as from different field of it. So with more enabling, right, you are seeing a lot of people are adopting the technology to complete their work, right? And yeah, as for your information, more than 90% of the sequence data available publicly, okay, or in the database you can find is actually based on Illumina technology to generate, which is the reliable and accurate data that people are referring. Okay, and as time goes, also Illumina has improved such that every labs, every skill that you have for your work, also there's a solution for you. Okay, so making everyone can now do the sequencing. It's just like everyone can fly now, everyone can sequence. Right? Okay, and then with Illumina technology, it's basically more than 40,000 papers using Illumina technology for their publications, which later I'll show you, share you some examples of those publications as well. Okay, globally, there's more than 9,000 of instruments globally available and running to generate all this data. That's why it's a really huge data, and people are now moving on to the big data era on the NGS, with the NGS technology available. Okay, yeah. So as mentioned just now, we have a very small scale up until a very large scale. Really the industrial output from a very small one like sequencing a few bacteria up until like sequencing a few human genome. Okay, this can be done within like 2 days time, and basically you can get a few human genome out easily. Okay, so the workflow is automated on the sequencing. It's like 5 to 10 minutes. Hands-on, then you are walk away, letting the instrument to solve everything for you on the sequencing part. Okay, so we have a different range. So if you want to have a system that run internally for your own project or internally for some of the screening work, right? Do let us know. We can actually have ideas on suggest you the best instruments for your needs. Okay. Yeah, and with the NGS methods, right, is that basically enable a lot of applications, right? With a focus more on the human part, microbe part. So today the content, the topic will be more focused on the microbes as well as more the microbiome part. So on the human part, I will not touch, but if there's an interest of that, we can also have another session as well. But today's basically looking at the microbes, and then with the NGS, basically it's not just on microbe itself, is beyond the microbe, which looking at the community of the microbe itself. Okay, so as a start, initially a lot of technology focusing on the whole genome sequencing, which are looking at a single isolate genome of a bacteria. Okay, so this one really comprehensively profile the bacteria in depth, okay, which tells all the information about the bacteria, what variants are they, what virulence factor, what resistance do they have, and what does this isolate different compared to the other isolate, right? So these are the capability of the whole genome sequencing on the microbes that can tell you. And there's more demand over that we know that bacteria, a lot of time they don't exist in one isolate or one type of bacteria. So basically bacteria exists in the community, and that's why there's a drive towards the needs of more applications to actually investigate or discover such information. Hence, there's these applications called Metagenomic, which later I will have more content focusing more detail. But in here, generally, these are the main 4 applications you will see on the NGS, okay, where this is a Metagenomic, which is focusing on all the genomes that are present in the samples, okay, be it RNA or the DNA, right? So means that when you do the extraction, all the microbes, right, are present in the sample will be lysed, and then all their nucleic acid will be released, and then we will want to sequence all those nucleic acid. Then it will tell you the contents of all the microbes as well as the gene present in the samples, and what role do they play into that environment as well, and that community. Okay, so this really gives you a thorough overview of what is present in the sample. And if you look into the RNA, then you will be able to see what are the activity that are carried out by these microbes in the environment that you have a sample. Okay, so and then there's also enrichment. Okay, enrichment is basically focusing on just a piece of information that you want, but in a more larger scale of it. Okay, so like maybe thousands of regions that you want to target. So you can use the enrichment method to capture the targeted regions and then get those information out. Okay, so these like enrichment methods has been like used in like human, especially like respiratory pathogen, where inside respiratory there's a lot of microbes, okay, or pathogen, right? A lot of time the symptom is very similar, so hence you have challenges on like correctly identify which are the culprit or the pathogen that causing the illness. So with this enrichment, you are able to just focus on those significant sequences that can help identify or characterize the pathogen. Okay, and then also on the amplicon. So the amplicon is really focusing on just a very small subset of the genome, okay, which to tell you certain information. So it's not really all information, it's very targeted information. And in this case, like let's say if you want to look at all the bacteria that are present, the taxa of the bacteria that are present in the sample, then the amplicon will be able to tell you that by picking one of the common regions that all the bacteria have, or like in this example as well, like in fungus as well, you can actually use the same amplicon method to profile the bacteria present, as well as to profile the fungus that present, fungi that present in the samples. Okay, so and moving forward for like really microbiome, a lot of preference is really on culture independent methods. Okay, and metagenomic and amplicon method is really the culture independent method where you do not need to culture, and we know that to culture bacteria, right, and from the samples that from a community, right, a lot of the time you have difficulty to culture all of them, and basically only like 10% or 15% that the bacteria that are present in the sample are culturable, okay, without any optimization. So if you want to get a lot of optimization, you need to work on to get those, and it's not comprehensive enough. So I mean, to have a faster information, right, skipping the culture method is really the way to go in the future. Okay, So in the NGS, right, why the high adoptions of the NGS is also mainly due to its simple workflow. Okay, it's really comprehensive, which you can remember after this as well, that just need to know like in NGS, it involves 4 main steps. Okay, first is really the sample preparation, depending what kind of samples. Okay, and then which are the extraction that are suitable, be it RNA or DNA, what type of sample, be it a saliva or be it like the plaque, everything, okay, or the swab. So these are depending on the sample, and then that will determine the respective suitable extraction. Okay, and then the next step is on the library preparation. So in this library preparation is like to convert the nucleic acid, okay, into the format where the sequencing machines can read. Okay, so in here, like if you extract the DNA, you have a double-stranded nucleic acid helix format. So there's a lot of them. Okay, but then the instrument, if you load into the sequencing machine, they can't read it. So we need to process a bit, okay, to make it into a format where the sequencing machine can take and read and give you the sequence of the fragment that you ask them to sequence. Okay, so and also in this library preparation process, like it involves like what kind of information you want to convert into the format. Okay, be it like the everything, or just the RNA, or just the amplicon part of it. So this is where it differs from different application to application. Okay, and then when it comes to sequencing, so sequencing is really, as mentioned, is automated already. Okay, that you just need like 5 to 10 minutes hands-on time, and then they're ready to go. And now even like the reagent cartridge, everything are like just like printers where you just load it in, then you can print. Same goes to sequencing. You just load to your sequencing machine, add the library or the samples, then you are ready to do the sequencing. As mentioned, it's really 5 to 10 minutes kind of a work. And then you can walk away and then wait for the sequencing to complete, which it will decode all the sequence or the fragments that you load to the machine, okay, and then give you the sequence, the raw sequence data of each fragment, okay? And then all this raw sequence fragment is just like A, C, G, and T, okay? And if you look at I, basically it doesn't tell much information unless you actually do some analysis to make sense of the data, as in like, okay, because we know sequences are unique, right? So what are the uniqueness that tells the differences, meaning that from organism to organism, from gene to gene, from expressions to expressions, all these informations are able to be extracted out, okay, through the analysis. And then this is why, as mentioned just now, BioEasy, sister company of Science Vision, is really helping our customers to really addressing the analysis part of it. What kind of information you want to get from this, and how you want to do that, and what are the significant findings from the data as well. So later in the examples, I will show you like what are the results that you probably can expect from such application. And then those are results, just examples, right? It may cater for different needs as well. So different projects may have different needs. So we can look into really specifically what you need, and then address accordingly. Okay. Yep. Okay, so that is on a quick go through on the NGS technology. So as mentioned, looking into the metagenomic, what is actually metagenomic? So it's really an approach that go beyond ordinary limits, okay, by looking into the genome of the organism. Okay, so now we know that every organism have their barcode, which is in the genome of itself. Okay, so this genome basically makes up the organism itself. So we human have 3 billions of bases that make up each and every one, which are unique, right? And to function normally. Same goes to bacteria. They have on average like 5 million bases in each of the bacteria, okay? And then by decoding them, you will know who they are, right? And then like if you look at a community, right? Whoever have this sequence, it will be unique to that particular organism. So in these cases where the research and detections all are, they basically complement each other. So with the technology, it's able to help you to characterize the pathogen or the bacteria. And then with that, you are able to use that information to help you analyze and ID the sequence if you found similarity of the sequence to that particular organism. So here, this is what metagenomic, looking at everything that present in the samples. Okay. So in metagenomic, right, there's two main approach. As mentioned just now, metagenomic is really a culture independent method, which you bypass the culturing process, which can be biased, okay, and also inconclusive sometimes, okay. And then it explore the genetic diversity, looking at really the all genetic diversity within the samples that you sample, okay. And then you also identify the population structure, looking at what are the dominant populations, what are the bacteria that dominant, making it like you know what are the progressive, which are the bacteria that actively working as well. All these informations is able to get from the metagenomic, and also to understand like the interaction of all these microbiome community by looking at the genes that present in the samples, okay, the activity that they transcribe, right, for their RNA work, right? So basically this will tell the activity within the samples, and that will indicate for you what does it mean, right? Okay, so I'll give you an example on later, some of the example to like how to interpret from those data and how this data actually help them to do some conclusion. Okay, so in metagenomics, like as mentioned just now, there's two approach. One is targeted sequencing, which focus on particular regions. Okay, for this one, you are looking really at the clade specific, or really looking at the taxa only. So meaning that like for bacteria, if you look at all the bacteria that are present, but you weren't able to know what genes they have, but you can only predict based on the bacteria that are present, the genes that they have in the sample. Okay, so targeted sequencing is really just focusing on the taxa. So the information needed is not really high, okay, but the information that you can get, I mean the data needed for this targeted sequencing is not high, but the information you get also will not be high. So in NGS, right, the more data that you have, right, normally it will translate to the more information, or vice versa. The more information you want, then you will need more data in the sense. Okay, so another metagenomic approach is called shotgun metagenomic, which this is really sequence all the DNA and RNA that you extracted or collected from the sample. Okay, so with this information, right, you are able to tell the taxa. Not only the taxa, it's also telling you the relative abundance as well as the function part of it. Okay, so what gene is present in that, why that gene is needed. So you from there you'll be able to have clear pictures why this bacteria exists, and because due to this particular gene. Okay, and then it also span across different organisms. I mean different taxa, from bacteria, archaea, fungi, virus, and even protists. So there, I mean, depending on how you do the sampling and extraction, what information you will get from there, we will be able to know. Okay, and then with that, right, and this is what the information you need today, okay, to be able to describe the taxa in the many samples, okay, and then what are the average community between the samples, and also the variation between the samples, like healthy and disease samples, right? What are the differences? Okay, and then also compare the community, different state, meaning that like different stages or different populations, right? Different diet, right? All these are able to change the microbial community, so making it like really unique, okay, to observe and tell the information. Okay, yeah. So 16S metagenomic is really the most common application, because it's a straightforward, easy, and then simple approach that you can adopt. It's just like a few PCRs, then you can go for the sequencing to get the information out. Okay, and then these are the workflow, and you now basically have demonstrated the success of this workflow, and then helping a lot of the metagenomic researchers have getting some answer from here. Okay, so how it works is like we know all the bacteria that have 16S RNA region, and in this 16S RNA, there's a consensus region as well as a variable region. So in the variable region is where these sequences different from bacteria to bacteria. Okay, by characterizing these sequences, then you are able to tell this sequence belongs to which bacteria, and then we can also count, right, how many copies of these sequences present in the samples that you collect. Then we can do an actual community profiling on the proportionals part of it. Okay, yeah. So as mentioned, this workflow is simple. Okay, and then it's really you can search a lot of publications out there has been using these methods to do. It's really a focus on the local specific, which you can just pick one region and change to another, like 16S. May not give you, this is example of V3 and V4. You can pick like other regions, V1, V2, or you can pick other like V9 regions for your own work. So it's very flexible in the sense that, and then this also not just limit to bacteria. So in fungus, if you have any other region, like example here, ITS, then you can also just change the target and then to focus on metagenomic those region now. Okay, yeah. So and these are the workflow for the 16S metagenomic, where you see here it's very simple as mentioned. Sample prep, meaning that you are doing the extractions, okay, accordingly. These are example looking at the soil example, okay, where you pick up the V3 and V4, but however the workflow is still the same across any kind of sample. Okay, and then go through the library preparations and then do the sequencing and get the analysis done to get the result out. Okay, so these are really a streamlined workflow. So changing the different targets will really just changing on this here, as well as like the extraction, because like if you extract from the fungus, then you will just focus mainly on the fungus itself. Okay, so that's why, as mentioned, these are the really simple workflow has been streamlined. You just need to modify a bit, then you can actually focusing on other type of aggregate. Okay, yeah. However, with that bear in mind as well, although it's simple, it's really cost effective, that there is some limitations to the approach, right? In here, like sometimes it's impossible to really design a primers that really capture all the bacterial profile or the microbes profile that you want. Okay, so in this case, like some variable region of 16S are able to differentiate this group of bacteria, but some cannot. So that's why it's not really like perfect in the sense that you capture everything. So that's why you need to decide as well what kind of a bacterial community that you are looking into it as well. And then also the sequence error is sometimes inexcusable for the real taxa. So we need to really set some limits of appropriate limit of detection for this particular approach as well. So that's why you need to validate and evaluate the approach before you really go for the large scale. Okay, and then species level identification not always possible. So for some of the variable regions, right, they can only differentiate up until like genus level, right? A species level is really beyond, because there isn't significant differences to differentiate between this species and that species that under the same genus. Okay, and then relative abundance is really not absolute, because like they involve amplification, right? So the amplification may have some changes to the relative abundance depending on the performance. So that's why some evaluation is needed to know how much is deviated. Okay, and then lastly, as mentioned, PCR bias on the relative abundance. So some of the consensus region which the primer binding to have a less efficiency as compared to the others, so making such issue for the PCR bias. Okay, then and because it's enabling, as mentioned, like sequencing is really affordable day by day. Okay, so making it like a lot of people are going into the shotgun method, which is everything like the metagenomic part of it instead of the targeted metagenomic. So they want to look at everything of it. So and also like there's evaluations on the shotgun method, which is like a shallow shotgun method, which you do not really need like 50 million reads to get the profile like the 16S. So you basically can go with like anything like 5 million reads of a bacterial profile to really capture the same profile as what in the sample. So making it like now a shotgun method is also one of the cost-effective way, okay, which will reduce a lot of bias that you may have for the epicon basis approach. Okay, and here you see here with even like very little data per samples, they are able to differentiate the sample type accordingly, because when you have different profile of the microbiome, it indicate that these samples belongs to which category. Okay, so with that, right, they are able to group accordingly based on the microbiome profile that are found in the sample. Okay, so that's why it's really powerful in the sense that even with little data, okay, you are able to do such differentiation, and it's really clearly distinctive between those significant different type of it. Okay, so more information if you want to do how to evaluate this, also you can look into these papers they are elaborating every aspect for the this shotgun method. Okay, and then you can look for that. Okay, and then the advantage of having shotgun metagenomic is really much more than what 16S metagenomic can do. Okay, it's, I mean, as mentioned, all the metagenomic approach is really culture independent method. Okay, and then it really gives you the relative abundance, really reflecting as actual, because there's no PCR involved. Okay, in the process, so really what you get is what you see in the sample. Okay, and then metagenotyping. So with this more information, more gene function, everything, right, it's actually help you to do like metagenotyping, such as like what to identify the gene loss, gain, everything. If there's a mutation, all these things also in the gene as well. Okay, and then like it's really hypothesis free, where you do not need to design anything. Okay, like for the targeted sequencing, metagenomic, you need to decide which region you need to do. But here it's really just extract, and then you can proceed with the sequencing. Okay, it involves pathogen detections. Okay, it can also potentially have like enzyme function present in the samples to know what it is. And then really this one really can go down until species, and in some cases we do see at the strain level detection is possible as well, which I'll show you some example later on. Okay, and then you can actually have more samples to put in, because that is very cost effective now. Then you can actually do a really large scale project on a shotgun metagenomic. Okay, and then also looking into some of the metabolic pathway analysis with the genes function information that you can get. Okay. So another part of metagenomic is really looking at the RNA part of it. Just now the shotgun metagenomic is looking at the DNA part of it, but this metatranscriptomic is looking into the RNA part of it. So there is a lot of microbiome-based therapeutic companies has using metatranscriptomic to do diagnostic. Okay, and we also do see pharma companies are really embracing this pharmacomicrobiomic, okay, on how drugs is metabolized by the microbiome. Okay, and then like develop this chemical-based drug that are interested in whether they are modified by the microbiome in some cases, where if the bacteria is there, you can ask the bacteria to modify and then have that drugs delivered accordingly. So that's why it's like really like the treatment, if like there's bacteria there, then you will create the treatment if you take the drugs. Yeah. So on the metagenomic and metatranscriptomic tool already let, I mean, help a lot of companies, right, to determine what microbe and function present in the sample and compare that to healthy and also disease samples. Then they are able to do such diagnostic as well. Okay, and then this really approach that will gain insight into functional and complex community. So how this community work? What's the ecology between all these microbes, right? How do they coexist? How do they function together? And also host and microbiome interaction. All these informations are able to be decoded through this metatranscriptomic, because you know what activity, because although the gene is there, it may not be transcribed. So that's why looking into the transcriptomic part of it, you will be able to profile like gene expressions across the transcriptome. Okay, so you get some insight into the gene and the pathway that are being expressed, and also compare to the expression profile under the different conditions such as like healthy and disease. Okay, and also I find out like some of the novel features information as well. Okay, so these are the quick overview of what metagenomic is all about. Okay, and that will be like really all this metagenomic is really looking to profile, okay, or discover the microbiome part of it. So here I will give you some of the example of the microbiome discovery, which can be enabled by the NGS and for your reference. Okay, yeah. Okay, so with NGS, basically is really enable researchers to easily interrogate a microbial community, because we know that the more microbes or bacteria present in the sample, then that community become complex, right? I mean, there's a lot of interaction in between here and there, between the microbial community as well as the host environment as well. So with NGS, it's really catered for a lot of needs into like studying the microbial community, as well as like it enable a lot of different scale of study for that. Okay, so the first projects that is looking into the microbiome research is really the human gut microbiome project. Okay, where we know that after years of studies, right, we do see like certain microbiome really contribute to the disease. Okay, and then that's why like there's also treatments available based on the microbiome as well. So these are really progressive, and we do see also other related samples that focusing on the microbiome also benefiting the approach. Okay, and you see that, I mean, even microbiome has been really called as a second genome, because it actually influence the human health. Okay, so by looking at the second genome, which is the microbiome, it will really tells you the health condition as well as what are the situations that you are having. Okay, and because it can help identify potential disease association, as well as like looking into the microbiome based therapeutic. If you have certain disease that are associated to microbiome, then potentially there will be some therapeutic solutions for that. So we do see a lot of progress when the microbiome research takes off. Okay, more and more solutions are made available now for that. So you can look furthermore onto those solutions in a lot of reference. So in here, okay, I won't go too much, but if you have an interest, we can actually have a more discussion later on as well. Okay, and then that is on gut microbiome, and we know also that oral microbiome is really the second largest and diverse microbiota after the gut. Okay, which can be sometimes having more than like 700 species of bacteria in the sample. Okay, so that's why it's also really important to study the oral microbiome, okay, which it can give you a lot of information and indicator for you. And that's why also human oral microbiome database also being built up for this purpose. Same goes to like human gut microbiome, which their profile can be different, okay, because like in oral, right, there's conditions are really different from the gut. So basically, they have different profile in a sense. Okay, and then after years of research, we do see also oral microbiome also review the health condition, okay, of the oral, especially I believe guys, if it's on this dentistry segment, you will be more familiar on like what are the diseases that are really caused by bacteria and what are the bacteria that are causing those disease. Okay, so I mean, not from the research itself, it's not only limited to just the oral disease. Okay, there's also more towards on other diseases as well, such as obesity. Okay, if you see that there's certain growth or increase of microbiome bacteria and decrease of certain bacteria, right, that indicates the disease occurrence, right, such as diabetes, liver disease, pancreas cancer, and also colon cancer as well. So this there is a lot of findings that indicate the disease to the oral microbiome. Hence, oral microbiome is really an important areas or information that we need, okay, in order to maintain or to make sure that the health conditions and with that, right, or if that is the case where it can indicate the health condition, right, then oral microbiome is sometimes really a more how to say non-invasive way of studying this or testing or screening this disease. Okay, just like COVID, right? I mean, there's also non-invasive way. You just take your saliva, then you can able to detect it. So these are the things that if this is possible, right, then we will see more non-invasive detection is enabled. Okay, so looking at the importance of a microbiome, oral microbiome, we do see like a lot of work, and basically I will categorize all this work, right, focusing on different approach like targeted metagenomic, shotgun metagenomic, as well as the metatranscriptomic. Some of the examples, there is a lot of them. I just pick a few which can help you to understand better. Okay, so first is on the 16S metagenomic, which is a targeted sequencing approach. You will be able to see like they are looking, this is a really large cohort study, okay, with 1,300 samples focusing on this one, and then they have basically come out with a significant finding that first of all, saliva and the blood community structures and microbiome are different, okay, and also like they also do see the correlation between the microbial species and the severity of the periodontal. So in these pictures here, right, showing that the healthy, medium, moderate, and also the mild, moderate, and also severe condition of the periodontal disease. So from here, these are example of the two causing bacteria. Okay, you can see here that for the healthy one, they basically have very minimal of these bacteria, but for the severe one, they really have much more than the healthy one. So same goes to these Cellonomas bacteria as well, which show the same profile so this one, because the group is really huge, right, this is really quite conclusive that it's not just individual cases, but it's really the whole population or the whole general disease of this. Yeah. So more, I mean, I just picked some interesting finding to share with you. What can you expect from metagenomic? And then, I mean, for in-depth, you really can refer to the papers for more information. Okay, yeah. So another example of the 16S metagenomic I want to share also is looking at like this small scale of metagenomic study, which they focus on like looking into the periodontitis status, okay, between the health and disease of an older woman, okay, because they believe that some of the hormone changes also will due to this disease, but they also want to look into like whether bacterial changes is significant or is it caused by anything. So this is somehow a preliminary study where the richness of the bacteria, right, is actually associated to the disease, right? For the healthy one, you basically have less rich diversity, but for the disease one, you will see much more diversity at the sample. So that is really telling you that some information about whether is this profile indicating disease or the health of it. So there are really specific communities of organisms that are associated with a periodontal and other healthy periodontium. Okay, so and then also these are also related to the aging information as well. Okay, so these are a preliminary finding. Okay, more deep one you can actually look into the paper. Okay, so this on the amplicon sequencing and amplicon metagenomic. I want to share also like on the shotgun metagenomic, which is really more and more researchers or work have been focusing using this approach, mainly because like now cost is really reduced significantly. Second, also the information you can get and the conclusions you can get from here is really a lot. Okay, and then yeah, so you see here, just looking at a small scale study between like 23 children with caries, as well as 24 children with healthy dentition. Okay, so and using the shotgun metagenomic, and the data requirement is really not that high. Okay, like it's just like 5 to 30 million read per sample. Okay, and then with 2 samples, really putting a lot of data to see the differences. Okay, so here you can see already like they have managed to list out, okay, if like there's a healthy condition, which are the pathogens actually abundance are higher, okay, as well as like if you towards the carries the conditions, right, then like what bacteria is present in more abundance, okay. So from here, in future, I believe if you can profile or identify the presence, the abundance of those bacteria in the sample, that already can help you do some conclusive screening on what's the conditions of the patient, okay, having these caries. So and what stages are they, and then how do they overcome such disease? Okay, yeah. So that really a comprehensive looking at even small scale of it, but it's really providing you a lot of information from there, and then looking at what genes are in there that contributing to these caries. Okay, as I know, some of the caries will have formed this biofilm or what. So these are the activities of that particular bacteria that carry out. Well, okay, yeah. So and another significant short-term metagenomic example I want to share is also looking into one type of I believe you guys are more familiar than me. One kind of like inflammations or a disease for these implantations of the tooth. Right. So basically, they look into 3 groups. Okay, one group is really healthy implants. One is like there's some symptom, and another one is really the term is like peri-implantitis, which is, I think, a severe kind of a situation. So they look into the shotgun metagenomic profile, and then the middle one, which the implant with a mucositis, is really sit in between. But the distinct differences between the healthy and the peri-implantitis is really obvious, as you can show here. Okay, you can see like the healthy one is grouping in one areas, and then the peri-implantitis is another group, and then like the richness is different, okay, and then all the informations are really different. So you can see the healthy group have different profile as on the peri. So really is somehow quite conclusive, like if in future you get the sample profile we fall into which category, then you can immediately conclude the condition as well. Okay, so the microbiome-based classifier really can accurately predict the clinical peri-implant condition. Okay, and this is not just to tell it is or not, but it's also telling you the stages and the disease progression. So it can tell at what stages, is it mild, severe. So these are the conditions that they can indicate by using the microbiome as well. So as you see, using NGS to do such work, right, it gives you a lot more information than previously observed by eye or observed by microscopic kind of work. Okay, yeah, and some more is like really getting a very small sample out of that. Then you can actually look into the really big pictures of the disease condition. Okay. Yeah, and the last application is on the Metatranscriptomics. So I'll show you some of the example here. Here, this is how they carry out the work, right? I mean, there's 2 to 3 biological replicates of mRNA libraries per growth stage. Okay, while there's 2 biological replicates for the DNA. So the MG stands for the metagenomic, which is a shotgun metagenomic, looking into the DNA. But the MT is looking at the RNA part of it, which can tell you the activity and the transcription happening in the samples. Okay, so they basically here you can see in here, right, and this is they decrease the pH as it goes on the time frame. So when they reduce the pH, right, there's a bloom of bacteria, okay, that grow happily. So meaning that these bacteria like low pH environment, so hence they will tend to grow, and because of that also. This is causing a disease where you see this is a carcinogenic lactobacilli. Okay, when you the pH drop. So I mean that's why I think acidic condition is really promoting the growth of this bacteria. and then like it also able to like indicate the transition point of many bacterial gene expression activities. So these are the activities that you can see by looking at that. Yeah, if you look at here, the black bar is actually looking at the DNA. The gray bar is looking at the RNA. So if you look at the DNA itself, you won't know the activity. You know they're present, okay, but they do not know whether they are active or not, okay. So with this metatranscriptomic, you are able to know is that bacteria active, okay. If they're active, normally the expression level is really high, and then from there you will be able to capture. So this is why this information indicate like when the pH getting lower and lower, this pathogenic, this carriogenic bacteria is actually growing. Okay, sorry for the terms here. So these are the informations you can extract from the metatranscriptomic. So I'll just capture some of the interesting findings for your reference. Okay, and then also, as mentioned just now, a lot of information has pointing that the microbiome can be served as a diagnostic tools. Okay, so in here, okay, so so here you can see these are using for the cancer. Okay, so here they basically look into the sample itself, where they pick up like a few groups of the samples, okay, normal controls. So basically from here they can see like the this blue color is the control, the orange color is the oral cancer samples, okay. So from the saliva metatranscriptome, okay, they already can group accordingly to the disease, okay, oral cancer, and also the control. So indicating like this can be a potential diagnostic indicator for oral cancer. And they even do a model performance on all this cohort, okay, and they achieve like 90 up until like 97.9% specificity in some cases, okay, where it really can differentiate an oral or give an indicator for oral cancer, okay. Hence early treatment can be done for the treatment. So with that, right, it can help to prolong or even like cure this oral cancer, as I understand from this publication. So here on average they need very little data, which is 1.5 million reads that map to the RNA. So even with that little data required, you basically can already do like a detections or indicator screening to check whether is it an oral cancer potential or not. So that's really a powerful and impact. Okay, and then and for Illumina instruments, like even the smallest instrument can easily generate 4 million sequences in a run. So that's why 1.5 million is really not a lot. Okay, so the highest instrument basically generate billions of reads for your work. Okay, so with that, right, I'll come to the summary of the information I have, right. In summary, right, metagenomic approach is really listed, summarized in here, where you will see like shotgun metagenomic, metatranscriptomic, as well as a targeted epitope metagenomic. Okay, so this is really to study the microbiome part of it. So like, let's say if you want to look into any sample, right, then you can determine what information you want. Okay, whether it's just a taxa, if it's just a taxa or the bacterial profile, right, then you can look into the targeted epitope metagenomic, which is focusing on the metabarcode region, such as like 16S or IDS. But if you want to know what gene functions are present in the samples and a more comprehensive, higher resolution down until species or even strain level, then you can go for the shotgun metagenomic. And then if you want to know the activity of the organism, right, then you need to look into the metatranscriptomic of the work. Okay, so that's basically come to the end of my presentation. Hope I have shared you some insight into how NGS enable the microbiome work. Okay, and then hopefully this can be some of your knowledge gained after this sharing session. Yeah.
Okay, thank you very much, Mr. Lim, for a very informative session today. Very interesting and eye-opening. At least for myself, I am clearer on the difference, especially between targeted sequencing and metagenomic and shotgun. Metagenomic, which is up until now it's a little bit confusing actually.
But those are more on the technical part. I mean, that's why, I mean, generally, I just want to give an overview how people use different applications for different work. So if you really need a more like technical, or we can really look into that. Yes.
Yeah. Thank you very much. Right. So we are open for questions at the moment. I have one question from Prof. Chen Yang. So how much bioinformatics support can you offer to the researchers here?
Okay, so I mean, different researchers have different needs. So some will just want to like outsource services. I mean, like they want to outsource the analysis. Some will want to, because I mean, they have a large scale research, hence they want to like do it more cost effective. Then they actually can get hardware tools set up in-house to really run it internally. Okay, and then also I mean, sometimes also like depending what your need, we can actually have a solution to that. Like let's say you want to customize a certain analysis, right? We can have a consultant to talk to you, what can you do? So really, you can name it, we can try to resolve it. So as mentioned, we do help train up people, okay? We help set up, we actually help establish the pipeline, and we even develop software if you have a needs on that. So basically, like let's say if you have a lot of samples, right? Then and the particular analysis you want to do, right? Just let us know, okay? We will see which are the best option for your consideration. Yeah.
Okay, thank you so much for that. Any other questions perhaps from audience? Okay, okay, while waiting, hopefully there will be some soon. I actually would like to ask, because to be honest, I am at the moment quite involved in doing this kind of research, and what I found is it's not many company or such who are quite understands about oral microbiome related research. For example, what we are usually having problem is mainly the sample collection, because for example, like dental plaque, for example, in healthy patient, it's quite minimal compared with healthy. I mean, compared with disease. So it's a bit difficult sometimes to get a good DNA yield. So in terms of sample collection, for example, what do you usually recommend?
Okay. So for that's why I mean that's why there's two situations. If really you have very challenging on getting sample, the metagenomic is actually one of the way why people adopt that is also partly the sample challenges where they can't get like enough yield for them to do. But with the Ampicon method, right, you are able to amplify even though it's very minimal. Let's say even less than 1 nanogram, right? You are able to amplify or capture the profile or the taxa out of that. Okay, but in the case that you are able to get more than that, like let's say 10 nanogram, okay, then shotgun metagenomic is always their preference. So in terms of sampling, right, I do come across some of the recommendations from people who focus on this research. Maybe I can share with you later on, like how people do that and what are the recommendations they have for that. I mean, for us, as long as extraction can be done and then the yield can be measured, then we were able to advise you which are the best approach. Okay, but in terms of like how to collect that, then I think those people which are in this field, especially I do come across some of the publication and literature which summarize all this. Okay, I think that there's at least two I can think of, and then I probably can share with you how they do that and what's their recommendation. And because we know that sometimes you get barely enough, should you proceed or not, right? I mean, we do hear from the customer when it comes to how best they can. We don't want also like later on it's not conclusive, then I mean it's a waste of effort as well. So yeah, really there is a publication on recommending how you carry out or design your experiment in these areas. I can share with you later on. Yeah, I mean my email is stated just now in the slide. Just reach out to me, Lixin at Science Vision, or you may reach out to Mr. Vincent for the respective information as well.
All right. Thank you very much. Thank you so much.
Yeah, just to add on, the shotgun metagenomic is now in, I mean, if as long as you can get like 1 nanogram, you are able to do the shotgun metagenomic. Yeah.
Okay, that's very I mean, interesting to know. Yeah, and very good. Right. So do we have any other questions? No, so far. Okay.
So Dr. Husna?
Yes.
Yeah, so this is Vincent. Okay. Let me just show you my face as well. All right. So I mean, so for all the participants or for attendees, if let's say you have any questions, please feel free to drop us an email. I think the flyer there already, we have the email and also the contact number as well. So feel free to drop us to let us know what is your inquiry about. We are more than happy and then to further discuss the case with you, because we treat and we always think it's about that each of the project actually is a unique one. So we cannot have a one general solutions to cater for all. So that's why try to let us know what is the challenges that you're facing. So we will sit down together with you and then we solve it together.
Sure. Thank you so much for that, Mr. Vincent. Okay, so far I think if there is no other questions, then again, as Mr. Vincent has already mentioned, and also Mr. Lim as well, they have shared the email address, so please feel free to approach them in regards to your project. Anyway, I thank you again, Mr. Lim, for the talk. Yeah, and I think we shall conclude the session today for now. Yeah, and I would like to thank everyone again for your attendance today. Yeah, and with this, I think I thank you and have a good day, everyone, and do take care of yourself. Okay. All right.
Thanks so much.
Yeah, thank you for having me to share this. I mean, we have more sessions.